Hi everyone, welcome back. In this lecture, we'll learn about the structure and function of skeletal muscle. We'll begin at the level of whole muscles, working our way down to smaller and smaller scales. We'll see how muscles generate force at the molecular level, how contraction is regulated by the release of calcium, and even what happens to muscles when you die. All right, let's get her done.
Climbing up on Salisbury Hill, I could see the city light. Wind was blowing, time stood still. Eagle flew out of the night. He was something to observe. Came in close, I heard a voice, standing, stretching every nerve. Had to listen, had no choice. I did not believe the information. Just had to trust imagination. My heart going boom, boom, boom. Son, he said, grab your things, I've come to take you home.
Let's begin our study of muscles with an overview of what they do and some of the key characteristics that allow them to carry out their tasks. By weight, muscles constitute the largest of the four tissues, about 40 to 50% of body mass. Humans have 640 different muscles, but luckily, you don't have to know them for this course. Muscles control movement, stabilize body position, regulate the volume of certain organs, and move substances. Cardiac muscle pumps blood through the circulatory system, and smooth muscle propels food through the GI tract. The least obvious function of skeletal muscle is heat production. Most often, the purpose of muscle activity is to produce motion, and heat is wasted energy. In shivering, where skeletal muscles begin to shake in small movements, heat is the intended product and is used for warmth. When the core body temperature drops, the shivering reflex is triggered to restore normal body temperature. Like neurons, muscles are excitable cells, meaning they can receive stimuli and respond by conducting action potentials. This causes them to contract, shortening along their length and creating a force that can be used to do work. Muscles are extensible, meaning they can be stretched to a certain extent and still contract. Muscles tend to return to their original resting length due to elastic protein components in the muscle fibers. Mammals have three kinds of muscle: skeletal, cardiac, and smooth, that can be distinguished by their shape, the number and position of nuclei, the presence of striations, and whether they are under voluntary or involuntary control. Skeletal muscle has elongated cells with multiple peripheral nuclei and visible striations. Most skeletal muscles are under voluntary control. Cardiac muscle has branching cells, a single central nucleus. They have striations, and they are under involuntary control. Smooth muscle has spindle-shaped cells, a single central nucleus, no striations, that's why they're called smooth, and smooth muscle is under involuntary control. As we start to look at the structure of skeletal muscles, it is important to keep in mind what level we are looking at, because muscles are like tubes within tubes within tubes, and it's easy to get confused. Muscle cells are called muscle fibers due to their elongated shape, and they are grouped into bundles called fascicles. Control of skeletal muscle is primarily voluntary, but some muscles like the diaphragm and postural muscles don't require conscious control. They work without having to think about it. All parents intuitively know this. That's why they'll say, "Go ahead," if one of their children threatens to hold their breath until they die. Skeletal muscles are an orderly arrangement of contractile cells and connective tissue. The contractile cells, called muscle fibers, actively shorten and generate large amounts of force. Connective tissue surrounds and infiltrates all aspects of the muscle, providing strength and support. The entire muscle is surrounded by a layer of connective tissue called the epimysium. Epi means outer. Bundles of muscle fibers, called fascicles, are surrounded by a connective tissue layer called the perimysium. Nerve fibers and blood vessels run between the fascicles on the outside of the perimysium, as indicated in the diagram on the right. The endomysium, which is essentially the same as the basal lamina, surrounds individual muscle fibers. All these different connective tissue layers are continuous with each other and with the tendons that connect muscles to bones. So the connective tissue as a whole provides structural support to keep the muscles from tearing themselves apart. The endomysium, which surrounds individual muscle fibers, has one additional, less obvious function. It acts as an electrical insulator between muscle fibers. Why, you might ask? Well, skeletal muscles, like neurons, are excitable cells, meaning that they conduct action potentials, which are essentially electrical impulses. If you recall from high school physics class, electricity running through a wire induces a magnetic field surrounding it, which can induce a flow of charge in a neighboring wire. The same principle works in muscle fibers, which are in close proximity. Muscle fibers don't all conduct action potentials simultaneously. Some are working while others are resting, which helps to delay the onset of fatigue. This insulating material makes sure muscle cells only contract when we want them to. A mature skeletal muscle cell is an elongated tube called a myofiber. You may have already noticed that muscle cells and their components have special names with prefixes like myo and sarco that basically mean muscle or muscle-related. For example, myofibers have a plasma membrane that is referred to as the sarcolemma, and many nuclei called myonuclei. The myonuclei lie just inside the sarcolemma. You can see them in the cutaway drawing of a muscle fiber at the top left. This is because the interior of a myofiber is packed with the contractile elements, the so-called thin and thick filaments, bundled into structures called myofibrils that occupy most of the space inside the cell. A small number of muscle precursor cells, called satellite cells, are closely associated with mature muscle fibers and are found just outside the sarcolemma. You might have picked up in the last slide that I mentioned that mature skeletal muscle fibers have many nuclei. It's true. A myofiber is a syncytium, meaning it contains multiple nuclei. In this case, due to the fusion of many myoblasts, muscle precursor cells, each with a single nucleus. This process, called myoblast fusion, takes place in two waves during embryonic development to form mature muscle fibers. Once they have formed, muscle fibers no longer divide, meaning they are postmitotic and terminally differentiated. That is, they begin to express genes involved in their final function. They can't go back.
Thank you, Doc.
No, son. Thank you.
Oh my God, you can't go back. You can't go back, can you?
It's all right.
I'm sorry.
It's all right. I best be getting on home before Alicia begins to think I got a girlfriend.
The microscope image on the left, taken from my lab, shows multinucleated myotubes. The nuclei are stained purple, that have differentiated and express a protein called myosin, shown in red, that the individual myoblasts don't have. You can only see their nuclei. Because muscle fibers don't divide, the number you have is set at birth and is the same you have as an adult. But we are bigger than when we are born, so how do muscle cells grow? Postnatal growth occurs as a result of hypertrophy, an increase in muscle fiber diameter due to an increased number of myofibrils. The increased size of myofibers results in more forceful contractions. Hypertrophy can also be induced by resistance training. Muscle fiber lengthening occurs by adding more of the minimal contractile units called sarcomeres, which we'll learn more about shortly. Both of these processes reflect an increase in protein synthesis in the muscle. Adult skeletal muscle has a remarkable ability to repair after injury. Skeletal muscle regeneration involves the activation of satellite cells, a population of muscle stem cells that are stimulated to divide by factors released by the degenerating muscle fibers. They proliferate, differentiate, and fuse to form new functional muscle fibers. Some cells do not commit and remain as muscle stem cells. These satellite cells also contribute to postnatal growth and hypertrophy. Satellite cells are closely opposed to muscle fibers and reside within the endomysium or basal lamina. Skeletal muscle cells have several specialized subsystems that regulate muscular contraction. One of these is called the transverse or T-tubular system. T-tubules are finger-like extensions of the plasma membrane that dive deep down into the interior of the muscle fiber. As you can see in the diagram, they occur at regular intervals and are connected to each other along the width of the muscle fiber, but not along its length. A section of T-tubule surrounds each myofibril and is flanked on both sides by thickened regions of the blue structure, like a sandwich. The three structures together form a triad. Triads can be isolated biochemically and their component proteins purified and identified. The function of the T-tubular system is to carry the action potential, which travels along the sarcolemma, down into the interior of the muscle fiber. Note that the fluid inside the T-tubule is ECF, because it is continuous with the outside of the cell. The sarcoplasmic reticulum, or SR, is a modified endoplasmic reticulum that surrounds each myofibril like the sleeve of a loosely knit sweater. Like the T-tubules, it is connected across the width of the fiber, but not along its length. The SR is thicker near each end next to the T-tubules. These regions, called the terminal cisternae, store calcium ions inside. As you'll see, calcium ions regulate muscular contraction, and the SR is stimulated to release calcium in response to signals from the T-tubules. Myofibrils are composed of an array of thin and thick filaments, which themselves are made up of the contractile proteins actin and myosin, respectively. The overlapping arrangement of myofilaments within a myofibril creates a pattern of alternating light and dark bands, the I bands and A bands, respectively. The A band corresponds to the length of the thick filaments, and the I band is the region between the thick filaments where there are only thin filaments. This pattern is repeated along the length of the myofibrils, and the myofibrils are all in register with each other, and so give skeletal muscle fibers their striated appearance. On closer inspection, more features are apparent. The electron micrograph at the bottom right shows a lot more detail. In the middle of the lighter I bands on either side of the darker A band is a so-called Z line. It is also called a Z disc because it is actually a circle, but looks like a line when seen from the side. A sarcomere, which is the minimal functional contractile unit, extends from one Z disc to the next. These units are repeated along the length of the myofibrils. In the middle of the A band is a dark line called the M line, for middle, and on either side of the M line is a lighter area that is collectively called the H zone. Use the diagram on the left to see how the myofilament arrangement creates these different zones. This end-on view shows how the thin and thick myofilaments are arranged along the diameter of the myofibrils. Each thick filament is surrounded by a hexagonal array of thin filaments, and each thin filament has 3 thick filament neighbors. It's a little harder to make out in this electron micrograph due to fixation artifacts. Depending on where you take the cross-section through the muscle fiber, you get a particular pattern of thin and thick dots. Notice that a section through the M line shows the thick filaments are connected by proteins that hold them in register with each other. What do you think the pattern would look like if a section was taken through the Z disc, indicated by the arrowheads? If we zoom in even closer, we can see the proteins that make up the thin and thick filaments. Thin filaments are composed mostly of actin monomers, called globular or G-actin, that polymerize to form filamentous or F-actin. This actin polymer consists of 2 rows of actin monomers that form a twisted filament. Actin is one of the 2 contractile proteins in skeletal muscle. Thin filaments are also composed of 2 kinds of regulatory proteins. The rope-like tropomyosin molecule wraps around the actin filament. Tropomyosin has a complex of 3 proteins, the yellow spheres in the diagram, called troponins bound to it. As we'll see shortly, tropomyosin and the troponin complex regulate the interaction of the thin and thick filaments to control muscle contraction. The thick filament is composed of myosin molecules. Each myosin molecule is shaped like 2 golf clubs twisted together. The myosin tail, twisted golf club handles, point towards the M line in the center of the sarcomere. Tails of neighboring myosin molecules lie parallel to one another, forming the shaft of the thick filament. The 2 myosin heads, the golf club heads, are attached via a flexible neck region and project outward from the shaft in a spiraling fashion, each extending towards one of the 6 thin filaments that surround each thick filament. The myosin heads are the working parts of the thick filaments. They interact with actin to cause movement of the thin and thick filaments. Hugh Huxley and Jean Hansen, who did their postdoctoral work at MIT, were one of the first groups to use electron microscopy to study biological specimens. They found by comparing electron micrograph images of stretched, resting, and contracted muscle taken just prior to fixation that the relationships of the thin and thick filaments changed. Based on these images, they proposed the sliding filament theory of muscle contraction in a landmark paper published in Nature in 1954. Prior to that, most scientists thought the actin and myosin proteins stretched and recoiled to change length. This animation shows, in a single sarcomere, how the thin filaments slide relative to the thick filaments. A sarcomere is the smallest functional contractile unit. Shown next are 3 sarcomeres and then 6 sarcomeres. Notice the difference in length of the 6 sarcomere unit when it is stretched versus when it is fully contracted. Skeletal muscle fibers have hundreds of sequential sarcomeres, so they shorten considerably over their length. The natural resting length of our skeletal muscles maximizes the ability of the muscle to contract when stimulated. If the resting length is shorter or longer, contraction is compromised. The effect of resting fiber length on muscular contraction is referred to as the length-tension relationship. The length-tension relationship refers to the amount of tension or force that a muscle can generate at specific starting lengths. This phenomenon can be explained by the sliding filament theory, which says that the tension produced is proportional to the amount of overlap between the thin and thick filaments. The greatest tension is developed when there is an optimal overlap, which luckily just happens to correspond to the resting length of our muscles. At shorter and longer lengths, less overlap produces less active force, and the relative positions of other proteins that make up the sarcomere interfere with contraction. After several decades of studies, the biochemical mechanism of muscular contraction, known as the cross-bridge cycle, shown here schematically, was fully revealed in the early 1970s. We'll look at each of these steps more closely, beginning at the top, but you could start anywhere since this is a cyclical process. At our starting point, the myosin head has a molecule of ATP bound to it. The myosin head is in its low energy conformation at a 45-degree angle to the thin filament and is not attached to actin. The myosin head has an ATP-ACE activity, meaning it can hydrolyze ATP into ADP, an inorganic phosphate. The ADP and inorganic phosphate remain bound to the myosin head for now. The energy from the high-energy ATP bond is transferred to the myosin molecule, causing it to adopt a high-energy conformation, and is now 90 degrees relative to the thin filament. Myosin is energized and ready to bind to its binding site on actin, indicated by the purple ellipse. This is the state that muscles are in at rest, primed and ready for action. The energized myosin head now binds to the binding site on actin and forms a cross bridge between the thick and thin filaments. In this diagram, the M line at the center of the sarcomere is to the left. Myosin binding to actin causes the ADP and inorganic phosphate to be released. This causes a further change. The myosin head rotates towards the center of the sarcomere, reverting to its low energy conformation, 45 degrees relative to the thin filament. However, because it's still attached to actin, it moves the thin filament relative to the thick filament. Because this step produces a force and movement, it's called the power stroke. Another molecule of ATP binds to the myosin head and detaches it from actin, bringing us full circle, and the cycle can continue. This slide provides an overview of the contraction process. Just read it through and try to take in what it says. We've just seen that ATP is the power behind the cross-bridge cycle, but calcium released from the SR calls the shots. Recall that the sandwich of 2 terminal cisternae surrounding a T-tubule in the center is called a triad. We can zoom in to see how these different compartments associate with each other. The diagram on the right shows the three compartments are closely associated and run parallel to each other, with a small gap between them. Voltage-gated calcium channels in the T-tubule membrane, called dihydropyridine receptors, because they bind the drug dihydropyridine, span the gap and physically attach to ryanodine receptors in the SR membrane. Ryanodine receptors, which bind the drug ryanodine, are calcium release channels that, when opened, allow calcium ions to flow through. This cartoon shows a different view of a T-tubule and a terminal cisternum, and a close-up of the DHP and ryanodine receptors spanning the gap between the two membranes. Notice that the two proteins are physically connected. Let's see how this works. An action potential travels along the sarcolemma and down the T-tubule. The voltage-sensing portion of the DHP receptor, indicated by the plus signs, changes the conformation of the protein in the membrane in such a way that it pulls on the ryanodine receptor and opens a channel, allowing calcium ions to rush out of the SR into the cytoplasm. A good analogy is pulling the chain on the bathtub drain plug, which allows the water to run out. However, we still haven't seen how calcium regulates contraction. In resting muscle, the cytoplasmic calcium concentration is low, about 10 to the minus 7 molar. The myosin binding sites on actin are covered by tropomyosin, preventing the myosin heads from binding. The opening of calcium release channels in the SR allows calcium to flood out, raising the cytoplasmic calcium concentration 1,000-fold to about 10 to the minus 3 molar. Calcium binding to troponin C causes a conformational change in the troponin complex, and that change is transmitted to tropomyosin, which shifts position, exposing the myosin binding sites on actin, allowing cross-bridge cycling to occur. This animated video shows cross-bridge cycling. See if you can recognize all the steps that you just learned about using the key on the left to identify the molecular players. You might not recognize the silver cone. Magnesium is a cofactor used by the myosin ATPase to hydrolyze ATP. How does contraction end and allow muscles to return to the relaxed state? In the SR membrane, there are calcium pumps called sarco or endoplasmic reticulum calcium ATPases, or SERCA, that continuously transport calcium ions back into the SR. This is against the calcium concentration gradient, so it is an active transport process, and therefore ATP is required. These pumps are always active, even in relaxed muscle. They maintain the low calcium levels in the cytoplasm. They are analogous to the bilge pumps on boats that pump water back out if it has leaked in. When calcium is released into the cytosol through the ryanodine receptors, it overwhelms the capacity of the SERCA pumps and calcium levels rise. But like Dory says,
just keep swimming, just keep swimming, just keep swimming, swimming, swimming. What do we?
Or in this case, the SERCA pumps just keep pumping until the calcium levels return to resting levels. In the SR, calsequestrin and other calcium-binding proteins act as calcium buffers. These proteins help hold calcium in the terminal cisternae after a muscle contraction, even though the concentration of calcium in the sarcoplasmic reticulum is much higher than in the cytosol. It also helps the sarcoplasmic reticulum store an extraordinarily high amount of calcium ions. Each molecule of calsequestrin can bind 18 to 50 calcium ions. ATP has 3 roles in skeletal muscle contraction. 2 of them involve the cross-bridge cycle. Hydrolysis of ATP energizes the myosin head, which eventually refers to a low-energy conformation during the power stroke, and converts the energy into a force that causes movement. ATP also detaches myosin from actin, allowing the cycle to continue. ATP also powers the active transport of calcium ions back into the SR, keeping the cytoplasmic level of calcium low. If you search for images of rigor mortis, which means rigidity of death, you can find a pub, a band, a World War II era bomber, and even a few creepy photos of dead bodies. Immediately after death, muscles become flaccid because they no longer receive stimulation from motor neurons. Why do muscles become rigid after a period of time? We just saw that ATP is required for contraction and for relaxation of skeletal muscle, and for pumping calcium ions back into the SR. After death, a lack of oxygen means ATP synthesis ceases. In addition, cellular degradation begins, and membranes become leaky, allowing calcium ions to enter the cytosol from the SR. The elevated calcium removes the tropomyosin block, allowing myosin to bind actin. But since there's no additional ATP, myosin remains bound and no movement occurs, which fixes muscles in position. This begins at about 3 hours after death and lasts about 24 hours. After that, the actin and myosin proteins themselves are broken down and the muscles become flaccid again.
Imagine that you're watching your favorite crime drama show. A murder has taken place. The police and forensic experts are looking for clues to catch the killer. One of the most important clues to solving the crime is determining when the victim died. The expert looks at the body, checks the body temperature, and examines the body's position. After a thorough examination, the expert then states the approximate time of death. The question is, how does a forensic expert calculate the time of death? The time of death is not the exact time that a person died. It is instead a time frame in which the person could have died based on the evidence presented. Forensic scientists calculate the time of death using many techniques. Their first go-to method is to study the physical changes that happen in the body after death. So what are these signs? There are 4 post-mortem signs that tell a great deal about time of death. These are physical observable changes that occur in the body in the first 24 to 48 hours after the heart stops beating and the nervous system shuts down. The first sign, pallor mortis. As the heart stops beating, the circulation of blood throughout the body decreases. The organs farther away from the heart, like the skin, stop receiving blood. This gives a corpse the pale, lifeless appearance that we associate with death. This paleness, which appears 15 to 30 minutes after death, is called pallor mortis. It is one of the first identifiable signs of death. The second sign, algor mortis. Humans are homeothermic, which means that we maintain a constant internal body temperature. This constant average temperature is maintained by balancing the heat produced in the body with the heat lost. The circulatory system is an important mechanism by which humans maintain an average internal temperature of 37 degrees Celsius. Heat is produced primarily through the various metabolic reactions happening in our body. This produced heat is dissipated from deeper organs, such as the liver and brain, to the outer parts, such as the skin, through the movement of blood. Under the first layer of skin, the epidermis, lies a network of capillaries. These capillaries allow heat produced by the body to be released into the air. Without the heart beating, blood cannot distribute heat and regulate body temperature. This leads to the body temperature reaching equilibrium with the external temperature. If the temperature is less than 37 degrees Celsius outside in the first hour after death, then the temperature of the body will decrease by 2 degrees and then continue to decrease by 1 degree every hour. Algor mortis is not an accurate way to back-calculate the time of death, however, as it varies based on external conditions, such as weather, and is affected by whether the corpse was wearing clothes or not. The third sign, rigor mortis. This sign is one of the most important signs when it comes to determining the time of death. Rigor mortis, which literally means stiff death, is the contraction of the muscles in the body after death. Before rigor mortis, all the muscles relax in the step called primary flaccidity. This happens because, after death, brain activity decreases until it completely stops. Without the brain giving muscles a signal to contract, the muscles remain in a relaxed state. Primary flaccidity lasts for 1 to 2 hours after death. The first muscles to relax are those in the eyelids, fingers, and toes. In this stage, the corpse usually releases urine and gas, as muscles controlling urination and passing gas are relaxed. Primary flaccidity is followed by rigor mortis. Muscles stiffen up in rigor mortis because of cells being broken down by digestive enzymes. This cellular breakdown is a normal part of body decomposition and is called autolysis. The breakdown of cells makes holes in the boundary of the cell and its compartments. This releases calcium ions from the separate compartment within the cell where they are stored. This calcium is crucial for initiating contraction. During normal living conditions, a signal from the nervous system triggers the calcium to release from its compartment. Another factor causing rigor mortis is the lack of ATP, the energy-giving molecule in the muscle cells. Once calcium initiates muscle contraction, myosin, a protein molecule that moves things around in the cell, binds to actin, a long chain of proteins that form the skeleton of a cell. Once the myosin binds to actin, it uses the energy from ATP to pull the actin, making the muscle fiber shorter and shorter with each pull. Immediately after death, muscles can still produce ATP. Muscles have reserves of glucose in the form of the polysaccharide glycogen, which it breaks down in the absence of oxygen to provide ATP. This ATP is used up in muscle contraction, but the muscles also need ATP to relax. This means that the actin and myosin will remain contracted until the muscle is completely decomposed. Rigor mortis starts in the same order that primary flaccidity does, with the eyes and fingers, and slowly progresses to the rest of the body. Rigor mortis reaches its peak 12 hours post-mortem. After that, the muscles begin to relax in the same order that they began contraction. After rigor mortis comes a second cycle of relaxation called secondary flaccidity, which is due to muscle degeneration. Digestive enzymes break down the proteins in the muscles, including the muscle contracting machinery. Forensic experts use this progression of muscle relaxation and stiffening to estimate the time of death. They will check to see which parts of the body have stiffened and make an estimate based on what they find. It is more reliable than algor mortis, as it isn't as affected by external conditions. The fourth sign, livor mortis. After death, blood is subject to the forces of gravity, now that the heart isn't moving it around any longer. It pools wherever the forces of gravity is strongest. This starts about 15 to 30 minutes after death and starts to become visible 2 hours after death. Lividity, as it is called, starts to become fixed after 6 hours as the blood vessels begin to break down. After 8 to 12 hours, a purple coloration develops. This purple color develops because the deoxygenation of hemoglobin forms deoxyhemoglobin, which has a purple-blue color. Livor mortis can be very useful for determining time of death. Once lividity is fixed, it doesn't fade, even when the body is moved. If a part of the body came in contact with a firm surface that obstructs the blood flow, an impression of it will show on the skin that is surrounded by pooled blood. The color or pattern of lividity can allow forensic scientists to guess not only what the time of death could be, but also the cause of death. Lividity in the extremities, such as the fingers, would suggest that the body was upright, as opposed to lividity developed on the back, which would suggest a horizontal position. These four signs are commonly used together in order to estimate the time of death. The time of death is not 100% accurate, however, because of so many other factors, such as the environment, weather, clothes, or whether the victim was moved. Forensic experts will most likely give a time frame rather than an exact time. So, the next time you're watching a crime drama, don't forget to be that guy and point out all the loopholes you see.